Since we went over stains last class, I don't have to spend too much time reviewing that. Just to give you an idea, we have positive stains, okay, negative stains. Positive, they're usually positively charged. They attach to the surface of the microbe. So you can see the example of positive stain here. When the microbe is stained and the background is not. Okay? The negative stains are the ones that stain background rather than the microbe. So you can see microbe is not stained. Okay, the negatively charged microbe is not stained. The background is dark. The good thing about it, you sometimes can see the external appendages, like capsule, for example, or some other weird shapes that can exist on the surface of the microbe if you do negative stain. Of course, you're not going to see the fine structure of the bacterial cell. Very fine structure. Those stains are simple. So let me put it this way. If you would take your petri dishes that you grew colonies on, and you scoop 2-3 colonies of different color and put them in a wet mount and stain them, all cells would look the same. You would have, let's say you would add crystal violet, you would have blue bacilli, and you would have blue cocci. And you wouldn't be able to tell them apart except for the shape. Make sense? So that's the concept of simple stains. Concept of differential stains is that they let you to tell microbes apart based on certain principles. Stains, what do they bind, first of all? When they bind a microbial cell, what do they bind? The membrane, yes, the envelope. We'll call it an envelope because they have membrane and the cell wall. So they bind what's outside. So depending on the structure of that outside, you can differentiate the cells. For instance, gram stain allows you to tell apart cells that are gram positive and gram negative. I do not know if Hans Christian Gram, the guy who developed this stain, actually knew about those fine differences between different bacterial classes. It turns out this stain reflects the fundamental properties of the bacterial cell. Gram-positive cells have structure of the cell wall that is profoundly different from gram-negatives. And that defines their properties and to a large extent defines their clinical properties. Their resistance to various antibiotics, their resistance to antibacterial chemicals. That makes sense? So it probably was sort of an accidental, serendipitous discovery that actually reflects fundamental differences. Acid-fast stains are used to stain microbes which have so-called mycolic acid in the cell wall. It's Mycobacteria, that's why they're called Mycobacteria. They different, so you can see staining of Mycobacterium tuberculosis here. You have stains that allow you to identify intracellular structures like endospores. That's a big group of bacteria. They can form them. Endospores are the structures that help cells survive through rough times. Mostly survive through desiccation, through dry conditions. Flagella, we discussed that flagella are very thin. And flagellar stain makes flagella thicker, allowing you to visualize it, like in the case with this Bacillus cereus. And finally, capsular stain allows you to visualize capsule that surrounds the cell. Essentially, capsule is, as you can judge from the name, is a structure that protects the cell, okay, mostly from host immune response. Okay, so that's kind of an overview of staining. That's how we identify microbes that we just cultivated. The next question, the very tempting question, is there a uniform approach to cultivating microbes? How do we grow them? Are there things in common? And the answer is yes. There are common steps for pretty much every microbial organism that you can identify. The step of inoculation, step of incubation, step of isolation, and inspection and identification. I put them usually together. So when you did your lab with hand wash, what was the first thing that you've done? Took the cotton swab and did what? Swabbed your hand. You collected the specimen, right? What did you do with the cotton swab next? Yes, you streaked it on a Petri dish. You inoculated the Petri dish. Petri dish. Where did it go to? Incubator. So we incubated the sample, right? Then we pulled it out. We skipped the isolation step. We'll get to that. What you did next with the plate? You looked at it. Yeah, you inspected it, right? And with a little help from me, you identified the sample. You identified the microbes. You identified that there are two distinct, some of you had three distinct types of microbes on the plate. Okay, so that gives you an idea. You already did all those steps. And actually, what I plan on doing next week, we're going to do probably one of the most exciting labs in the course. As I mentioned, you're going to swab things, like whatever you want. Your cell phone, your purse, I don't really recommend the bottom of the shoe. It's just shitload of microbes. It's really a lawn of microbes and fungi growing all over the place. It's fun to see, but you can't really isolate the single organism. You can swab anything you want. And what I want to do next, it would be great if you would get a little notebook and just write down the results, because what I want to do when you isolate, when you sample the environment and you grow something on your plate, you're going to select the colony and you're going to study it throughout the semester. Does that make sense? So you're going to grow it. You're going to grow your microbe, one microbe. You don't know what it is, but you're going to work on it, okay? You're going to accumulate it in bigger amounts. You're going to you look at it under the microscope, you're going to stain it, identify whether it's gram-positive or gram-negative. You're going to check if you're going to do some other differential cultures to see if it's fermenting sugars or not fermenting sugars, if it's hemolytic or not. It's going to be a lot of fun. Seriously, it's going to be a lot of fun. So that's what I want to do. Okay? If you prefer to do it on, just on the separate sheets of paper and submit it to me in the form of lab report, fine. Because I'm going to keep them. I'm going to keep all those lab reports in a folder, so we can track down and see what you have isolated from the environment. Okay? Questions? Okay. So the first step, collecting the specimen and inoculating the specimen, inoculating the medium. Well, when you collect the specimen, one of the most important things is to collect it using the sterile instrument. Those cotton swabs were sterile, right? The syringe that you use to collect, say, patient's blood is sterile. The cup that is used to collect urine is sterile. You don't want to contaminate your sample with whatever you have on sterile instruments. So what can serve as the clinical specimen? In addition to skin swab, blood, or urine. Give me other examples. Fecal matter, good. Hair, yes. It will bear mostly skin stuff. Mucus, yes. Saliva. You can collect sweat. You can name pretty much any secretion of the body, especially external secretions. Tears, sure enough. You can collect lacrimal secretions. You can collect biopsy of the organ and see if there's any growth there. Okay, so in these terms, virtually everything can serve as the clinical specimen. Although you have to, especially with the fecal matter, it's kind of a tricky, because whatever's in the feces and whatever's in the intestine, those are not the same things. I'd be honest, because not everything that sits in your intestine gets carried away with the feces to the outside. It was shown on multiple occasions. But, you know, bless you, but the problem, if you want to sample somebody's intestines, it's kind of hard to convince people to undergo the colonoscopy just for the sake of little sample, you know, kind of a almost literally pain in the butt. So we have to stick to feces. Environmental samples. You will see that you can collect samples from built environments. Built environment is the term that is used for everything that we live next to, like table, faucets, doors, doorknobs, elevators, you name it. That's the built environment. Does that make sense? Okay, so that's where we collect sample from. We can collect water, we can collect soil. Again, we can collect animal feces, whatever you want. Some of the environments are very, very interesting to work with. If you would want to collect soil, not a big deal, right? You just go outside and collect soil. There's one microbiologist, and I always mix up. She's either at Kent State or University of Akron. She does studies on microbial populations of caves. She goes into the cave and collects samples. Caves are extremely isolated. You can imagine that. And she selects caves that are truly isolated, so people don't go there normally. You really have to kind of get out of your way to get into that cave. Here's the problem. If you go there, you must leave no microbial traces, which means you wear protective equipment all the time, not to protect yourself, but to protect the cave. And you have to collect all your human waste. All of it in special containers and move it out of the cave to avoid contamination by like human fecal microbes. So it's really interesting things, you know, that you have to do to really study microbial communities and leave them undisturbed. Now, when you collect the sample, you have to grow those microbes. Now, if you put microbes here, technically speaking, will they grow? They probably won't. Why? Too dry, something else. No food source, no nutrients, and it's too dry, okay? When there's a little or no food source, it is called oligotrophic environment. Essentially, caves are oligotrophic environment. There's not a lot of food out there, okay? So if you want to grow whatever is in your inoculum, whatever's in the syringe, whatever's on the swab, you have to put it on the medium. Medium is anything that can support growth of the microbe. It can be a mixture of chemicals like agar, or it can be a live organism like eggs or animals or plants. Even bacteria can serve as a medium for growing the viruses of bacteria. Does that make sense? Because without them, those bacteriophages are not growing. So you have millions of different microbial species. Do all of them grow on the medium in the lab? Why? Why not all of them will grow?
Depends on what they synthesize and what they use for food.
Sometimes medium is suboptimal, doesn't contain some essential factor, and the problem we don't really know. Okay? So, say you collect sample from your skin, you put it in the agar, you have white and blue colonies, you have two types of microbes there, right? Does it mean that there are only two types on your skin? Not necessarily, right? Maybe some of them just didn't grow. Okay? For instance, anaerobic bacteria will not grow on that TSA plate. They're going to die because of the abundant oxygen. So whatever anaerobes you had in the skin, bad luck for them. You see? Some bacteria become extremely tough to cultivate. You really just cannot put them out there and grow. People spend years trying to find the medium in the lab, medium that you can conveniently prepare to grow a certain microbe. And in the meantime, this microbe can cause diseases, can be identified using other methods, but you just can't grow it in the lab. Does that make sense? Now, medium, you see the illustration here. It can be a agar plate. You can grow them in the blood. You can grow them in animals, as I mentioned, but it doesn't have to be those three types. There are more media that we're going to chat about. Medium can be of different kinds. We can classify it based on the physical state, based on the chemical composition, and based on the function. And medium can serve different purposes. Like this agar plate, the TSA plate that you used, its purpose was to just allow microbes to grow. Almost all microbes will be able to do that. Other media will restrict the growth of certain microbes and give preference to others. Some types of the media will help you to tell apart different microbial species based on their unique features. So we're going to talk about how media can be classified, and yes?
What does assay mean?
Media that, so you inoculate media and it allows you to identify, say, multiple identification of species, precise identification of species. We're going to do the, essentially the assay media. You can say this and this, differential media and assay media, are similar in terms of the function. For example, there is the special test, the assay, that is called enterotube. So it's a tube that contains several little cells, wells. And each well has a specific type of agar with chemical indicators. When you inoculate this tube, you spread your microbe, your inoculum, through all cells. And the microbe starts to grow in all those cells and causes some chemical changes in those media. Or does not. And when it does cause changes, you can detect this by the color changes. and you end up with a certain code, like numerical code. And we're going to do that. We're going to do that lab. It's pretty interesting. So numerical code that you can then take a special catalog and put this code, look up this code in the catalog, and identify what you had as your inoculum. And usually it works pretty well. It allows you to identify it very, very closely. So that's going to be a say. Okay? Did I answer? Based on the physical state. So the one that you worked with, the agar, okay? TSA agar is solid. Okay? It has several very, very important features. You can see the example of the solid medium here, but nobody uses it in this form, honestly. I mean, what's the point to put agar in the tube? Come on. Everybody puts it on the plate. Okay? So the microbes grow on the surface, form colonies. That's a very important identification feature. Color. I'm going to take this lab that we did and use it as an example. Color. You had yellow, you had white. You already can tell apart two different species. Some of you, I think some of you did have that. The large white colonies, like big ones. That was bacillus. It looked completely different. So colony appearance helps you to identify the microbes. Actually size sometimes. If you tweak microbe just a little bit, if it mutates, it can form same shape, same color, but smaller colonies. So it doesn't spread that much. Okay? Solid media that we use, agar, is liquefiable. It's kind of an awkward English word. I don't think that actually, something like that. I'm not sure about my own spelling, so trust me, you're not going to get any points away for not spelling it properly. The idea here is that if you take agar and you warm it up to about 55, 60, it's going to melt. What usually is done, all the components are mixed together, thrown in a flask, water is poured over it, they placed in the autoclave. Autoclave is right behind that wall, right there. Placed in the autoclave, you run the program, it heats it up to 120 degrees Celsius, which is something like 250 Fahrenheit. And the whole thing melts. So when you pull it out, you have a transparent orange-brown liquid. And then you put it here and you wait. And when you can touch it with your bare hand without burning your hand. Not that you can hold it comfortably, but you can do pretty much this. It's time to pour the agar into the plates. You gotta move quickly because it starts to solidify. Actually, you can pour it when it's hotter. We did that because we added antibiotics, so you have to give it to cool down so antibiotic does not die. But you can just pour agar in the plates, leave them on the table, they will solidify with time, and you got your plates. You can do the same at home using gelatin. Jello, right? The main component is gelatin. It's liquefiable. You can heat it up, it'll melt. Why don't we use gelatin for bacterial growth? Any ideas?
Doesn't it have a high trace bacteria to make it?
What?
Doesn't it have bacteria already in it?
You can autoclave it. You can sterilize gelatin. I mean, bacteria can eat it. That's the problem. I mean, you inoculate your gelatin plate, you come over and there's like a swarm of bacteria and no gelatin. They ate it all. So it kind of sucks. Agar is not eaten by bacteria. That's the upside. So we usually use agar. Semi-solid. Kind of clotty. So if you will If you will lean it downwards, you see it's like a little bit of a liquid part here. If you would lean it over with the top of the tube facing downward, eventually it will very, very slowly leak out. Okay, it's like a gel. Why do we need it? Mostly motility. What does that mean? So this is the example here. So this is your control. Transparent, semi-solid, medium, nothing happens. You inoculate it with a non-motile bacteria. You see? You see that trace going down? You see that? This one. So that trace is where you inoculate it. You inoculate it. I don't have that thing here, but imagine the loop without the loop. Just pretty much like a needle. So pierce it, pull it out. Wherever you pierced it, bacteria stays there. It's non-motile, it doesn't move. Okay? Now if it can move, it can swim, usually if it has flagella, it spreads all across the medium. And you can see that medium is turbid. Turbid means cloudy, right? All across the medium. That makes sense? Well, this is kind of the cherry on top of the cake. Okay? It's so-called, so you can add something. In this case, it's sulfide ion in the medium. Some microbes can reduce sulfur. When they reduce sulfur, they essentially form this ion. Medium contains iron as well. Iron and sulfide ion form this compound. It precipitates, gives the black color to the medium. So this is example. I'm going to introduce some terms and we're going to learn them over and over again. This is an example of differential medium. You can differentiate motile and non-motile, right? And you can differentiate sulfur reducing versus non-sulfur reducing. You see? You can combine different features in one medium that allows you to kind of group bacteria. Liquid. Well, water-based. We don't grow microbes on ethanol. You're going to die. So water with nutrients. Okay? When we say liquid, it means that it's going to be liquid. It will freeze if you put it in the freezer. But other than that, temperatures above freezing, about 32 Fahrenheit or 0 Celsius, it's going to stay a liquid. We're going to add nutrients there. Nutrients can be precise chemicals. Which nutrients would bacterial cell need based on your knowledge of biological processes? Glucose, so carbohydrates, usually glucose, yes. Sometimes you can add some others, sugars. What else? Water. We can consider it as a nutrient. Depends. Lipids and carbs are sources of energy. Fats in the call it? Water. It's kind of a tough task. Forgot about something else. Protein. Well, not protein, but amino acids. Just supply them with amino acids. Some of them will need vitamins. So you can measure exactly, well, salts and stuff, but you can measure exactly all the chemicals that will support the growth of the microbe. Such medium will be called define. We'll get to that. Or you can just take brain. like a cow brain, and treat it with some enzymes like trypsin to break down proteins, and then dry it, the brain. And then when you need the medium, you can dissolve that brain powder in water. You got your nutrients. No, seriously. There's the beef extract, okay? Oh, come on. You cook liquid medium at home to make soups on the stock, like beef stock or chicken stock. That's your liquid medium. Leave it outside for 3 days and see bacteria grow. Okay? So that's pretty much what you do. Milk, perfect example. Leave it open outside. Trust me, there's going to be plenty. Try and drink it, and your intestines will serve as a detection mechanism. You can add indicators to it. Okay? They will change color. So here's the examples. No inoculation, and then you've got bacteria that digest urea. When they digest urea, they break down, they have urease enzyme. They release ammonia. Ammonia increases pH. That gives pink color. It's probably phenolphthalein in the medium. It gives pink color to the medium. So you can detect urea digesting bacteria. Okay, and a lot of bacteria that digest urea can be found in, surprise, surprise, urinary tract. Because it's a good place to digest urea. Any questions on the media states? Not really? Yes?
Viruses?
We'll get to that.
Okay.
We'll get to that. Give me 1 second. Composition. We kind of discussed that already. So this is the chemically defined growth medium. In one of the classes, somebody asked, do we have to know all components? No, you don't. Okay, no. You just have to understand the difference between the synthetic and the complex medium. In synthetic or defined, you know all the components to the letter exactly. You go in the lab, you have cans on the shelves. Can says alanine, glycine, vitamin A, potassium chloride, whatever it says, chemical. And you weigh a certain amount of each chemical, you throw it in a certain amount of water, you mix it, you dissolve it, this is your medium. You know exact concentrations. Okay, you see? Exact concentrations. Right? So that's your defined. It's convenient to grow organisms that you are familiar with. Like this Staph aureus here, you know its nutritional requirements. This is a complex medium. Brain-heart infusion, BHI. Pretty much you have like a powdered brain, powdered heart extract. You throw it in the water, there's a bunch of stuff. Fats, carbohydrates, amino acids, peptides, whatever. Probably some growth factors, you can only imagine. Do you know the exact composition? No. Like a chicken stock. Do you know the exact chemical composition of the chicken stock when you cooked it? No, you don't. Okay, but you don't care actually. As long as bacteria grow, it's good. Does that make sense? For as the source of nutrients, animal extracts, plant. So TSA stands for trypticase soy agar. So soy protein is being broken down. Okay? Whatever. Peptone, yeast extract, whatever you can think of that contains all essential amino acids, all 21 amino acids, can support bacterial growth. The question that can arise easily, why don't we use complex All the time. Why bother with defined media? Yes? Hmm? It's dirt cheap. Like seriously, those nutrients, well, not dirt cheap, but it is sold. You can buy like 10-pound cans of that powder. It's not a problem. Yes.
You maybe need to know the exact, precise way that you made that media
Why? You're on the right track,
for
but
growing that specific type of bacteria, like a recipe.
it will grow on the complex media just as well.
Scientific method if you like need to know the exact way to do it, or so someone can replicate what you just did.
Okay. Yes.
With complex, there's too many variables when you're trying to go through the lab.
So,
I was gonna say you can
go ahead.
find ways to kill it.
So if you take a nutrient out, is it gonna grow? Is that right? So say you have 20 amino acids and you want to know if that specific microbe can synthesize tryptophan by itself. You take tryptophan out. You know 100% there's no tryptophan there. Make sense? So that allows you, for instance, you want to select for the mutants that can grow without tryptophan. There you go. Make sense? On complex, you're not going to take out any amino acid. You just won't be able to. You see the difference? But complex, of course, is convenient to support the growth of many microbes. So say if you want to test fecal microbiome, and there's a ton. It's estimated 1,500 different species. You just throw some complex medium and hope for the best. Yes?
So does complex mean that it's an inaccurate lab report then?
Say again?
Like if you're doing this in a lab,
Yes.
would it make it less accurate? Would it make it inaccurate necessarily? I mean, dependent on what you're trying to observe from the microbes, right?
Well, it depends on your aim, right? But I mean, if you say, okay, we want to look at microbes of give me an animal. Cow intestines, okay? In a fecal sample of a cow. We don't know what is there. The only way, we have no idea. So we're going to put it on complex medium. Here's the exact composition. We took 50 grams of BHI, 10 grams of peptone, water, and mixed it all together and just grew the microbes on it. Does that make sense? So any other lab can take the same BHI, same peptone. Same water is kind of harder, but assume same water, mix it up and replicate your result. Does that make sense? But you won't be able to say, we have microbes that can synthesize tryptophan, because you supply everything. Okay? Even the most picky microbes will grow on it. Does that make sense? So that actually is a segue into different purposes and different types of culture. So media that TSA that we used is generally considered to be a general purpose. So for everything. And as anything that is a general purpose, it's okay. You know, it's okay for everything, but it's not perfect for anything. It's okay for everything. Okay? Enriched media is the one that maintains the growth of fastidious microbes. Remember I told you, I will tell you about the word that you have to know. Remember, if there is something, I will stress it out. Know the word fastidious. Fastidious microbes are the ones that require some specific component to grow. Right? One of the examples of fastidious microbe is the gram-negative cocci from the genus Neisseria. I bet you all know at least one disease that Neisseria genus causes. It's gonorrhea. Neisseria gonorrhea is a causative agent of sexually transmitted disease. Neisseria meningitis is one of the, you've heard about meningococcal vaccine, right? That people get. So that's Neisseria meningitis. Neisseria is a fastidious microbe that requires an additional nutrient. And the problem is we don't really know what does it want. But what we know is it grows excellent on so-called chocolate agar. It doesn't have any chocolate. The blood, the sheep blood, is heated up, pretty much cooked, and is added into the regular TSA. So it gives it a chocolate color. And you can see the Neisseria colonies in this particular example. Okay? In many cases, so, okay, what is so special about the blood? What do you think? There's one component of the blood that really helps bacteria to grow. Yes, excellent. Actually, I always wanted to learn by heart the Kipling's poem, Iron, called Iron, Master of them All, but it's actually, it refers to the microbes. Iron is, I can't even stress how important it is for microbial cells. All right? When we run the fever, what happens to the levels of levels of available iron in a human blood when we run the fever? They drop, yeah. Iron becomes less available. It's the natural mechanism that our bodies have to restrict the bacterial replication in the blood. Okay? So iron is good. Here's the cooked blood. Here's not cooked. So this is so-called blood agar. The undenatured, good, fresh sheep blood is added to agar. Erythrocytes stay intact. Some bacteria can lyse red blood cells. This is called hemolysis. So some bacteria can lyse red blood cells, some cannot. Which ones do you think are worse for humans? Which ones are worse? Yes, hemolytic ones, yes. They're bad. You can see the example of hemolytic microbes. Well, that's alpha, that's beta, that's going to be alpha hemolysis. That's going to be beta hemolysis. There's some weird naming. Pretty much, some of them cannot do anything to red blood cells. Some of them lyse them incompletely. And some of them, it will be, we're going to do a lab about it. You will see it's astonishing. You just see the absolutely clear area. So there's no blood, it's just transparent. All blood is gone, all those microbes just completely destroyed it, yes.
Which one's that?
Hemolytic. The one that break down red blood cells. Ask your question again.
Which ones are bad?
Between the ones that can and cannot break down red blood cells?
I just don't understand what you're talking about.
Okay. So microbes, some microbes, can destroy human red blood cells. Does that make sense? Some cannot. So the question is, which ones, as a general rule, would be worse? Ones that can break down red blood cells. Does that make sense? Okay? So, by putting those microbes on the blood agar, probably didn't stress it out. By putting these microbes on the blood agar, okay, we can see whether they can destroy red blood cells or whether they can't. Is that clear? So we can say, okay, this microbe that we have here is destroying red blood cells. We have to be cautious with it. It's probably a human pathogen or potential human pathogen. This microbe here does not destroy red blood cells, so it's probably benign. Those two microbes that you can see because of the screen, if you will put Staphylococcus epidermidis on the blood agar, it will grow, but red blood cells will remain intact. If you take Streptococcus pyogenes, the causative agent of the strep throat, and put it on the blood agar, blood will be gone in 2 days. It is hemolytic microbe. It destroys the red blood cells. That's why it's infectious and can cause a lot of complications. Is that clear? Did I answer your question? Good. Culture. Culture can be pure. You can see different examples of pure culture here. Staph epidermidis, Mycococcus luteus. This is, I think it's Serratia moshensis. Okay, different types of culture. It can be, when you culture a microbe, you aim to get a pure culture. You want to get one species. Does that make sense? You don't want things to be mixed up. Culture can be mixed. That's what you got when you sampled your skin. Did you expect it to be mixed? Yeah, most certainly. You expect to have more than one species. So mixed is good. It's okay. That's what we expect. We expect mixed culture from samples of environment, for example. Now, contaminated culture is when you expect it to be pure, but something crawls in, like in this picture, when you have some unidentified growth on what is supposed to be a pure culture. Pretty much the difference between contaminated and mixed and contaminated, you don't expect the second or third species. Does that make sense? Any questions on the purpose, general versus enriched, and types of culture? Not really? As I promised, selective and differential media. Selective media. Select. When we say college has highly selective admission, what does that mean? Not everybody gets in, correct. Not everybody gets in. Okay? Same goes for selective medium. Not every microbe gets to grow on it. Okay? Even if that media is inoculated with it. That allows you to tell apart different groups of microbes. So this illustrates this here. Okay, so you have a general purpose media. Okay? So you see all the different colors like yellow and supposedly blue, but I honestly cannot think of any blue bacteria. I can think of green colonies, but not blue, right on top of my head. But let's say you have different colors, all different species. You put exactly the same sample on the selective medium, and only one species grows. Because medium contains something that prohibits growth of certain types of microbes. Does that make sense? That allows you to, well, isolate, but more importantly, identify certain groups of microorganisms in your sample. The example here is Staphylococcus. Okay? Bless you. So, Staphylococcus is quite a unique group of microbes they can grow on the high salt. 7.5% sodium chloride. That's a lot. That really is a lot. Yes?
Is that why it can grow on our skin?
Good question. Might be. Well, it's probably not why they can grow without salt, but it's probably because others can't. You see? That may be a case. It's a good question. Okay? So they can grow at higher salt than other microbes. So make a medium that contains a lot of sodium chloride. That essentially prevents microbes like, say, E. coli or Salmonella to grow in that medium. So you restrict it to Staphylococci. And then some of the Staphylococcus species can use the sugar named mannitol as the source of energy in the process of fermentation. Others cannot. So if Staphylococci ferment mannitol, they produce yellow colonies. If they don't ferment mannitol, the colonies will appear red. Does that make sense? So, say we would have Staph epidermidis and Staph aureus. Okay? Two species of Staphylococci. Can they both grow on sodium chloride? What do you think? I just told you. Can they? Yeah, they can. Okay, so they can grow. What about mannitol fermentation? Turns out this guy does not ferment mannitol. This guy does. So you can start recognize the species. Does that make sense? By changing color of the medium. Right? This is the list of various media. You don't have to know that list, and you don't have to know what we use them for. I'm going to give some examples that actually we're gonna use. For instance, we're not going to use tomato juice agar, but it's highly acidic, and it allows you to isolate lactobacilli, which can normally grow at the low pH. Those are microbes that can ferment milk and produce different derivatives of milk, like buttermilk or yogurt. Okay, so they naturally grow in the acidic environment. Macconkey Agar. This type of medium contains bile salts, which restricts the growth of gram-positive microbes and allows you to pretty much isolate only gram-negative bacteria originating in the intestine. Okay, Suborot agar. It's acidic agar that restricts the general growth of bacteria but promotes the growth of fungi. So you see, we're not talking about selecting one species out of thousands and thousands of various bacteria. It's rather selecting the group, okay, gram-positive versus gram-negative, okay? That makes sense? Differential. Now, here's the example. This is your general purpose medium on the left. So you see all the colonies look pretty much the same. You put the same sample on the differential medium, and colonies now appear in different colors. So medium does not restrict the growth, but rather allows you to differentiate between microbial species. That makes sense? It contains something that gives different colors to different media. Great example is right here. Okay, so this is called triple sugar iron agar. It contains three different types of carbohydrates. So this is non-inoculated. This is growth of microbe. Let me erase it actually. So I'm going to highlight it in a different way. This is non-inoculated. This is growth of microbe without fermentation. No change. All you have is the bacterial growth. Here, you see the change, right? pH changes, and the color of phenol red changes from red to yellow. And you can see a little bit of red on top. What does that mean? There's no growth on top, right? Why? Say again? Yeah, it's anaerobic. Top, this part, is exposed to the air, right? Anaerobes just cannot live there. They just die. They can't replicate. So they grow happily at the bottom. That's where they metabolize and ferment sugars. Okay? This is either aerobic microbe or aerotolerant anaerobe. So the guy that doesn't use oxygen but can grow in its presence. So you can tell apart. And finally, this is the sulfur reducing bacteria. The same example that I gave you before, okay? When microbes reduce sulfur, it produces sulfide ion. Sulfide ion binds to iron, forms the iron sulfide, gives the black hue. And you can see that this black hue is localized to the top. So now this is aerobic microbe. It grows only in a superficial level. Does that make sense? Or its biochemistry may change, so it does not reduce iron in the bottom, but does reduce it on top because of the oxygen availability. Okay? How important it is, the differential medium. This is the chromagar. You see the list of pathogens. E. coli, Klebsiella, Proteus mirabilis, Enterococcus fecalis, Staphylococcus saprophyticus, Staph aureus. Those are most common pathogens of urinary tract in humans. Males, females, kids, doesn't matter. Each species, which name is written on that agar plate, shows up in the color that its colonies show up on that chromagar. So E. coli colonies are pink. Klebsiella is dark blue, Enterococcus is light blue, and so on. So you can put sample, urinary sample, or sample from the urethra, okay? Put it on the agar plate and quickly diagnose which pathogenic microbes are found in the patient sample. And a little digression, I know we talk a lot about fundamentals, but you probably want some clinical perspective. Urinary tract infections, first of all, are very common. Second, they may contribute, okay, if there is an infection in, say, urethra or urinary bladder, where can it go? Yes. And if it goes to kidneys, and what happens to kidneys? Huh? Filter blood. Well, if they infected, that they're not going to filter blood that well. What's going to happen? Yeah, so toxicity. Huh? You may, if it, but what's going to happen to your blood pressure? Eventually. Yeah, it's good. And since kidneys underperform, GFR goes down, you develop hypertension, chronic hypertension. Does that make sense? And actually, there's no good evidence, like solid clinical evidence, but a lot of physicians suggest that urinary tract infections early in life in kids may contribute to the development of hypertension later in life. Because one of the major differences between the child and the adult is that the child, especially 3-4 years old, they can't really describe what's going on. So you have a child with a fever. No other symptoms, fever. In many cases, it's a urinary tract infection. It may go without significant symptoms, okay, like physical symptoms, pain or dysuria. And if it ends up in the kidneys, then it may contribute to the renal dysfunction later in life. Okay? That makes sense? So these are examples. We're going to talk about differential media. I'm going to say a few words about some of them and we'll take a break. These are examples of different differential media. The blood agar, okay? That's classic differential. It supports growth of pretty much everything, but it allows you to differentiate hemolytic versus non-hemolytic. Mannitol salt, we just saw it. It's selective, and at the same time it's differential. So it selects for staph and allows you to differentiate the species of staphylococci. Okay, Macconkey, we just talked about it. It is selective. It selects for gram-negative enteric bacteria. It turns out it allows you to distinguish between bacteria that can and cannot ferment lactose. A digression. Which ones do you think are better? The ones that can ferment lactose or the ones that cannot? The ones that can, why? Huh?
So you're tolerant, so you can drink.
The ones, well, it's one, but mostly your lactose intolerance comes from the lack of enzyme, lactate dehydrogenase. Bacteria contribute, but not that much. Now, yes?
I was going to say they're designed to feed on lactose. They want to target anything else.
What does that mean? I mean, they're not designed, kind of, yeah. Do you, not you personally, do we drink milk? So the ones that digest lactose, that ferment lactose, would you consider them to be normal inhabitants of the tract? Ah, most likely, yeah. Right? Because lactose always comes. And they're used to it. And if there's something that cannot ferment it, this is suspicious. This species probably comes from, some other place. It's not supposed to be there. Does that make sense? The guys that can break down lactose are usually components of normal microbiome. The guys that cannot, usually pathogenic. And we're going to do that lab as well. Okay? Well, we talked about triple sugar iron, right? Before we get to break and then we chat about viral, then we'll do the lab and so on and so forth. One thing that I want to mention about differential and selective. One thing that confuses people, students, in microbiology, bless you, is that there are rules that are fairly vague. You expect something to be rock solid. You get that description, you're going to get differential media, and then turns out same media can be differential and selective. Okay? So get used to it. Get used to the fact that every rule will be flexible. Okay? We will stretch it. So it goes. Some media are only differential, some media are only selective. But a lot of them serve both purposes. Does that make sense? Now for this, I'm not that hungry about examples for the exam, but I really want you to understand the difference between them. Not only on the level of definition, Okay? I mean, of course, you must answer the question, media that preferentially supports the growth of specific bacterial species and inhibits everything else is, and you pick selective, right? But I can give you an example of actual media, actual, say, selective media. Describe what it does, okay? So it, say, Macconkey Agar supports the growth of gram-negative enteric microbes and inhibits the growth of gram-positive bacteria. What would describe it best? Okay, and you get answers. So you have to recognize it's selective. Does that make sense? So that's that. You have to employ that thinking. We're gonna take a break, and when we will come back, we will do the I hope, fun lab.